Good afternoon. My name is Mara Alonso. This summer I worked on a project about the sequestration of dye contaminants from water. My mentor is Neil Rosenthal, and I was working at Dr. Bergbreiter's lab. First of all, dyes are everywhere. They are in the clothes that we're wearing, they are in packaging such as food and cardboard packaging, they are in cosmetics, in plastics, and all of the industries that use these dyes have some leftover colored wastewater. These are called dye effluents. The textile industry in particular produces the highest amount of dye effluents, and this is mostly because fabrics have a limited absorption capacity. I made a quick demonstration in the lab about this. I dyed a piece of cotton fabric, and after I dyed it, I have some leftover red water. This would be a dye effluent. According to the literature, dye effluents can be toxic, mutagenic, and when they are released into the environment, they disrupt the ecosystems. The current treatments that are being used to treat the dye effluents, the most popular one is an absorption method using activated carbon. Activated carbon is this black powder that is very porous, so it traps all of the contaminants from water. It is coal-based, though, which is a non-renewable resource. But mostly, the big disadvantage is the recyclability challenges, because to recycle it, it needs to be heated up to really high temperatures. Also, biological methods are being investigated, which use either enzymes, fungi, or algae. This is not really feasible in treatment plants, making it hard to commercialize. The approach that we are taking at the lab to treat dye effluents, it started by an experiment we did in the past where we separated carboxylate anions from water using a PaO and PIB co-solvents. So for example, we had in this container water, on the bottom there's water contaminated with a carboxylate anion. And on the top, we have PaO with PIB co-solvents. We mix it in the vortex and then perform a liquid extraction, and we end up with purified water on the bottom, and on the top we have PaO, PIB co-solvents, and the contaminant, the carboxylate anion. This gave rise to the idea of separating dyes. For example, there are dyes such as methyl red that have a carboxylate group. So then we started looking into anionic dyes. Like I mentioned before, there's dyes with a carboxylate group such as methyl red, carmine, and 4,5-dichlorofluorescein. But there's also dyes with a sulfonate group, and these are bromocresol, methyl orange, and Congo red, which has two sulfonate groups. And these are the ones that we were experimenting with. As the extracting solvent that we use to remove the dyes, we use PAOs, or polyalpha olefins. I use PaO4, which is a mixture of structural isomers containing 4 fully saturated carbons, which may look like this. Using PaO as extracting solvent has the advantages that it is non-toxic, non-volatile, which is not flammable, it is insoluble in water, which means that water won't get contaminated by the PaO, it can be recyclable, and it is very inexpensive. As the other co-solvent, we use functionalized PIB sequestrants. PIB is available in the alkene form, and it is commercially available like this. I use PIB 20 molecular weight 2300, and it is also non-toxic, insoluble in water, and inexpensive. So I start off with PIB alkene. I perform a hydroboration reaction to get PIB alcohol, then an Appel reaction to get PIB iodide, and with PIB iodide, I perform SN2 reactions, alkylation reactions, to get the cations that I'm looking for. And these have an iodide counterion. So the cations that I worked with this summer were cosamidosolium cation, diethylmethylammonium, and N-butyl imidazolium. All of these had iodide as a counter ion, which doesn't work really well. So we mix it with sodium carbonate, and now we have the same cation, but with sodium carbonate as a counter ion. And this is what we use to remove the dyes from water. So now the procedure that we follow to remove the dyes are first preparing the organic phase, which is a 0.025 molar solution of the PIB cation to PaO. I also prepare a dye solution in 0.1 molar aqueous sodium carbonate. What I do is I grab a test tube. Then I add 5 grams of the dye solution. In this case it's blue because I used a bromocrystal dye solution. Then I add zero point 0.5 grams of the organic phase. I mix it in the vortex, and at 30 seconds of mixing, I start seeing that it looks clear, but I do 2 minutes. Then I put it in the centrifuge for 15 minutes at 3000 RPM. And this is how it looks when I take it out on the centrifuge. The water has been purified, and I analyze this by UV spectrum. Now, I first tried all of the cations that I made. So these are the 3 cations, and I tried it with methyl red and methyl orange, which the only difference in structure is that methyl orange has a sulfonate group, and methyl red has the carboxylate group that we mentioned before. So here on the UV spectrum, we see the absorbance here. This is the absorbance that it had before being mixed in the vortex. And after mixing with cold solution, we see that almost all of the absorbance is gone, which means that this worked really well and the water was treated the best. Then the next cation for diethylmethylammonium, it still shows some absorbance, but it worked well. For N-butylimidazolium, it works the least. And we see the same trend for methyl red, which worked better than methyl orange. So then I tried removing dyes from the dye solutions with all of the dyes that I mentioned earlier. There are the 6 dyes, and I removed it with cold-soluble cation, which is the one that works the best. In all of the dyes, it got removed more than 99% of them. I also tried an experiment to test the capacity of the organic phase. With a bromocrystal dye solution, I followed the procedure that I mentioned earlier, but after the water has been purified, I take it out with a pipette and I add more of the dye solution. And I did this 10 times. Throughout the 10 cycles, more than 99% of the dye got removed. Finally, I tried an experiment with more concentrated dye solutions. I did this with bromocrystal with a 0.1 millimolarity and 4,5-dichlorfluorescein with 0.2 and with cold-soluble cation. In the case of bromocrystal, almost 99% of the dye got removed, while in 4,5-dichlorfluorescein it was 96%. The next steps for this project are figuring out how much of the concentration can we remove in a single batch. Also experimenting recycling the PIB cation through ion exchange with either sodium hydroxide or sodium carbonate. Also trying additional cations such as ammonium and plutonium cations and see if the structure has anything to do with the sequestering efficiency. Lastly, investigating with different molecular weight of PIB. Like I mentioned before, I use PIB molecular weight 2300, but there's also available 450 and 1000. Lastly, I'm really grateful to have been part of this REU program, so thank you very much to the REU program organizers, and also a special thanks to Dr. Bergbreiter and the research group, who were very helpful throughout the summer.